Kylie, you may begin.
Okay, awesome. Thank you. Hello, everyone. Just as people are beginning to trickle in, as you know, my name is Kylie. I'm one of the summer interns for Lundquist, and I'll be helping moderating this last lecture for the summer fellowship series. So today we have Mr. Hayden McSwiggen. He currently is a PhD student at the Translational Research Program at the Lundquist Institute for Biomedical Innovation at Harbor, UCLA. He obtained his Bachelor of Science degree in molecular biology and immunology at the University of Nevada in Reno in 2016. He currently works in the laboratory for Dr. Wei Yan, where he is using cutting-edge single-cell techniques to elucidate the epigenetic mechanisms involved in germ cell development. He is also interested in understanding how environmental factors encountered during one's lifetime, such as diet, stress, and toxin exposure, can generate epimutations that are inherited through subsequent generations. So Hayden and Mr. McSwiggen, if you would like to take it away and begin your lecture on single-cell RNA sequencing.
Thank you, Kylie, for that introduction. So today I'm going to talk a little bit about some of the cutting edge techniques that we're using in the lab to analyze transcriptomic data. And I will also touch on some of the single cell techniques that can be used to analyze other epigenetic factors such as chromatin structure. So this is kind of a representation of some of the recent advancements in transcriptomic analysis. So the traditional bulk RNA-seq studies will use bulk populations of cells to get an average gene expression signature across all cell types in the tissue. So one of the analogies I kind of like to use is this fruit smoothie here. If you think about each fruit as an individual cell, and you blend these up into a drink and drink that smoothie, you can kind of identify the more prominent fruit in that smoothie, but you can't identify each individual fruit that went into that smoothie. So with single cell analysis, you would be able to identify each individual fruit, and in the context of biology, you'd be able to identify each cell that is in the population of cells that you're analyzing. So with single cell analysis, you are dissociating cells into individual cells. So one of the drawbacks of this is that you lose spatial information. So spatial transcriptomic methods have been developed to retain the spatial information, and this allows you to capture gene expression profiles at near single cell resolution in the context of the tissue section. So why is this important for biology? That's because biology is tremendously complex. There's about 40 trillion cells in the human body. Each cell has the same genome, but each cell has its own unique epigenetic program. And each one of these cells has a unique gene expression profile that is translated into its own unique protein activities. So this is the basis behind what Dr. McCary talked about in previous lectures about how we can get different cell types and why the eye cells are different from the toe cells. So again, for bulk RNA-seq, these are the traditional RNA-seq analysis studies. You would take a piece of tissue, grind it up, and lyse all the cells at once in bulk. And this will give you an average gene expression signature across all cells. So you can look at multiple genes, but it will give you one data point, and that's the average gene expression. With single cell RNA-seq analysis, first you dissociate this portion of tissue into single cells, and then you process these cells individually in parallel. So this allows you to resolve the gene expression signatures in each of the cell types. And we'd like to kind of visualize this data with these plots that I'm showing here. So this is a two-dimensional reconstruction of the single cell RNA-seq data. And what you're seeing here is individual cells represented by these little spots. And cells that have similar transcriptomes will be clustered around other cells with similar transcriptomes. And cells that have different gene expression profiles will be clustered sort of further apart, so you can kind of identify individual clusters that represent cell types. So why what are the benefits of single-cell RNA-seq analysis? Well, first off, you can characterize heterogeneous cell populations, as I just mentioned there. You can identify all cell types in your tissue of interest. You can also identify rare cell types or rare cell states. You can also reconstruct cell trajectories and lineage relationships, which is important in the context of in the developmental context. And you can identify dynamic gene changes between individual cell types. And you can also uncover new cell markers and gene regulatory pathways that are important for individual cells. And this also allows you to profile healthy versus diseased tissues and organs to identify which cell types might be affected in certain diseases. So these are a few examples of some of the discoveries that you can do, that you can accomplish from using single-cell RNA-seq analysis. So on the left here, this paper identified a rare cell type that wasn't previously characterized before. And this is a progenitor cell population. So if you isolate this population of cells, put it into a dish, you can grow pancreatic organoids that will develop all the cells in the pancreas, and you can pretty much study pancreatic biology in a dish. The example here in the middle, they used RNA-seq analysis, single-cell RNA-seq analysis to profile 15 major organs in the human body. So eventually we'll be able to analyze all different cell tissue types in the human body and create databases that will have gene expression profiles for every cell in the human body. And then on the right here is a paper published by John McCary, and they profiled most of the cell types in the testes. And we did this in both human and mice. And we could look at the different gene expression profiles in each stage of spermatogenesis. So this is just a quick snapshot of the developmental history of single-cell analysis. The first single-cell RNA-seq analysis paper was published in 2009. And at this point, they were manually selecting cells. So this was a really long process. They can only analyze about 1 to less than 100 cells per experiment. And so many other ways of sorting cells have been developed. You can sort them into individual wells of the plate and do the analysis with multiplexing cells. You can also create nano droplets, just small reaction chambers that will sort cells, and you can analyze cells in parallel using this method. And so one of the more commonly used methods today is developed by the company 10x Genomics, which was established in 2017. So this is the method that we use in the lab today, so I'll kind of focus on this method. But they utilize this nano droplet formation to sort cells and then process up to, right now it's up to 80,000 cells in parallel. They just released a version that gets up to about 1 million cells. So it's showed that 10x Genomics was published 2017, and the next year after that, Science declared single-cell RNA-seq analysis the breakthrough method of the year. Nature kind of beat them to it. 2013, they declared it the method of the year, but nevertheless, ever since 2013, 2018, there's been an explosion in the number of single-cell analysis methods that have been developed. So I kind of just showed this here to show you that there's many methods out there to do single-cell RNA-seq analysis. But for this talk, I will just focus on 10x Genomics because this is the method that we use in our lab today. So how does this work? So the 10x Genomics platform uses this microfluidic device and creates these nano droplets, as I discussed previously. So what you're trying to accomplish here is sorting individual cells into these nano droplets so that you can process multiple cells at the same time in parallel. So you start off with these gel beads and your cells of interest, mix them together and hope to get one cell, one bead per nano droplet, or in 10x Genomics language, they call these gems, which is a gel bead and emulsion. And we'll look at this more closely in the next slide. But after you get a single cell in your gem and your gel bead into an individual gem, your cell will lyse, it will release RNA, and you can use reverse transcription enzyme to do reverse transcript, or you can use a reverse transcriptase enzyme to do reverse transcription. And then during this reverse transcription, your DNA will be labeled with a cell type specific barcode that you got from this gel bead. We'll talk about that more later. And then once you label the cDNA with your cell type specific label or barcode, you can pool these cDNA, finish creating libraries, and then sequence these libraries, and then do some data analysis further down the line. So this is a close-up look of these microfluidic devices. Here's the not close-up look, just what the chip will look like. So there's eight lanes here on the left. And this is one of the lanes in the video image here. So what's happening here is these gel beads are being flowed through these channels. You mix your sample in here, so your cells will mix with these gel beads, and then they'll pass through this oil channel, which will partition your gel bead and cells into a droplet surrounded by oil. So these are, we can refer to these as emulsions, and that's why they call them gel beads and emulsions. So looking at the gems more closely, you can see in each individual gem, what you're hoping to accomplish is get one gel bead, one cell, not two cells. A lot of the gems will have no cells, but that's better than having two cells because it's a single cell analysis. So once this occurs, the gel bead will dissolve. It'll release detergents that will lyse your cell, and that will release mRNA into this nano droplet that can then be used in the reverse transcription to convert your mRNA into cDNA. So looking more closely at the gel bead, on the surface of the gel bead, you'll have these little fragments of DNA, which we call oligos. And on these oligos, there's some important sequences. So one of the important sequences is poly-D-T, and this is just a series of T's that will allow the poly-A tail of your mRNA to attach. And this is the start. This is the start of the cDNA process, cDNA synthesis process. So when you're doing the cDNA synthesis, you'll incorporate this unique molecular index, or UMI, and this will allow you to identify how many RNA molecules you detected from each individual cell. You will also incorporate a cDNA or a 10x barcode, which is a cell type specific barcode. So later on, when you get your sequencing results, you can identify the specific cell type that the mRNA molecule came from. And the last part of the oligo here is just a read primer, which allows your mRNA to be sequenced later on in the process. So this slide can be kind of confusing or overwhelming, but I'll point out the important parts here. So this top left portion happens inside of the gem. You have your oligo that's attached, that's inside of your gel bead, and the mRNA that's released from the cell will attach this oligo. You'll perform reverse transcription, which will convert your RNA into complementary DNA, cDNA. And then skipping a couple parts here, you'll amplify the cDNA to produce enough products for sequencing, pretty much. And then through a couple more reactions here on the right, you'll add a few more sequences that are important for sequencing your library construct. But after all these reactions, you'll end up with a library construct that looks like this. And so what you have here is your RNA insert that's now converted into cDNA. You have your unique molecular index, which will help you identify how many RNA molecules you got from an individual cell. Your cell barcode, which will identify which cell all these RNA molecules came from. And then also sequences on the end that will help you during sequencing of this library. So after this, you can send your sequences to a sequencing company or however you want to sequence your data, and relax for a little bit until you get your sequences back, and then the real analysis starts to happen. So this is kind of how I like to look at single cell RNA-seq analysis and a lot of other analysis in biology, where the wet lab and the visualization or the figures that you see in your publication are really only the tip of the iceberg. The bioinformatics that goes on behind the scenes take a really long time. You can do this multiple times, and it's really important for extracting biological insights from your data. So for those of you that may have programming backgrounds or want to get into coding, I would strongly encourage that, because those skills are very helpful to have in a lot of fields, but especially with genomics fields. So this is just a simplified diagram of the typical steps used to analyze single cell RNA-seq data. So first off, you get your raw reads from the sequencer. You run it through a pipeline that will generate a count matrix, which is pretty much just a spreadsheet with cells across the top in the columns and genes here in the rows. And it will be populated by numbers pretty much that will give you expression values and this will tell you how highly expressed or lowly expressed each gene is in each one of these cells. So after this, you can do some pre-processing, some normalization, some QC, some filtering, and then you'll select the genes that are more highly variable, because these will be the most informative for your downstream analysis. So some of the downstream analysis that you can do, well, first off, you want to cluster your cells. So this will cluster your cells based on the similarity of the transcriptome. Similar cells with similar transcriptomes will cluster closer together than cells that have different transcriptome. And then you can look at the genes in each one of these cells to identify the markers of these clusters. And using those marker genes, you can identify which cell types are represented by these clusters. And then after this, you can do a number of other downstream analysis, trajectory, and you can construct trajectory analysis. And this will help you identify genes that are important for developmental processes. You can also do differential expression to identify genes that are important for individual cell types. So you can compare, in this example, these stem cells from these goblet cells and identify marker genes that are important for these individual cell types. You can also do compositional analysis to identify what cells might be affected more by certain conditions or diseases. So a few of the analysis softwares that are provided by 10x Genomics. The first one is Cell Ranger, which is a Linux command line based pipeline that will process your raw sequencing reads and generate the count matrix or the spreadsheet that I talked about a few slides ago. And then after that, you can import this count matrix into this Loop browser, which is developed by 10x Genomics, which is a visualization software. It's a desktop application with a user-friendly graphical interface. So this will allow somebody with no command line experience to analyze the RNA-seq data, look at the genes in their data set and which cells your genes are more expressed in. And you can also annotate these clusters based on those genes into specific cell types. So this is a great tool, but to get a further in-depth analysis, they have to go into some third-party software. And these are software that have been developed by the single-cell sequencing community. So the two most common ones are Seurat and ScanPy. Seurat is an R toolkit, and you can use this to pretty much do a lot of the same things as the browser that I talked about in the last slide, but you have more control over some of the filtering parameters and clustering parameters, and you also can produce different types of graphs and plots that you can't do with Loop browser. And these different types of plots are helpful for annotating your subtypes. And then for Python users, there's a Python toolkit called ScanPy, which can pretty much do the same things as Seurat, but you can also do trajectory analysis with ScanPy. And another benefit of ScanPy is that it takes less computational power, so you can process larger data sets with ScanPy. And this is important for when you're moving to hundreds of thousands or even millions of cells. This is going to be important. So beyond single-cell RNA-seq, I'll briefly mention the next evolution in single-cell technologies that allow researchers to analyze multiple modalities in the same cell at the same time. So I think you all know that there's multiple epigenetic factors that work in tandem to produce the observed RNA expression profiles. So being able to analyze these epigenetic factors and gene expression in the same cell at the same time can be very powerful. So this is the first iteration from 10x Genomics on this multi-omic approach. And using this method, you can look at open chromatin regions and link that data back to gene expression data to get a better understanding of the gene regulation in individual cell types. So the workflow here looks pretty similar to the single-cell RNA-seq, but before you sort your cells, you use an enzyme called the transposase enzyme, which will go into the nuclei and cut your chromatin in regions of open chromatin. So fragment your chromatin in open chromatin regions. And while it's fragmenting the chromatin, also insert these DNA fragments that you can capture using the multi-omic gel beads that I showed here. So you also have this oligo from the single-cell RNA-seq pipeline. And additionally, you have another oligo that will capture the fragments inserted by this transposase enzyme. So this method, you can more powerfully characterize cell types and states, which is happening in this figure here on the right. So you can see in this gene expression space, there is a cluster here that kind of looks the same, but you can't really tease it apart with gene expression database. But in the ATAC space, you can see that there's that these clusters here have distinct open chromatin profiles. And because all these cells are linked, you can use this ATAC space to separate these cells in your gene expression space. So it's a tool to help you identify which cell types are in your RNA-seq data. And then you can also identify novel gene regulatory interactions and interpret epigenetic profiles with key expression markers. So this kind of shows the analysis that you would do in Loop browser. So here we're looking at the expression of a specific gene. In this case, it's LEAF1, LEAF1. And you can see the expression levels of this gene here. And then when you look at the cell types where the expression of this gene is high, that naive cell type, the naive T cells, memory T cells, there's these regions of chromatin that are open. And so this is, you can identify this as a positive regulator of this gene. And then you can look at regions in the genome that are close to leaf one in the cells that have low expression of this gene. And you can see at this chromatin region, you have large peaks, but in these cell types, the expression is pretty low. So you can assume that this region is probably involved in repression of this gene. And then the other benefit of doing multiple modalities in the same cell at the same time is that you can maximize precious samples. So you don't have to split your sample into do two different analyses. You can do the same analysis at the same time. So building upon this, researchers are trying to see if they can identify as many modalities in the same cell at the same time as they can. Here's two kind of interesting techniques called T-seq and dogma-seq. So in both of these protocols, they use the 10x multiome assay that I talked about here, but also incorporated some antibodies so they can identify proteins in the same cell also. So I think this is kind of magical, and I think in the next couple of years, increasing the modalities and increasing the number of cells that we can process will allow us to gain a lot of cool insights into what's happening at the single cell level and integrating a lot of these epigenetic factors to really understand the whole system approach to what's going on at the single cell level. So I think single cell RNA-seq analysis is pretty cool, but there are some limitations. One of the limitations is low throughput, so the amount of cells that you can analyze at one time, although it is getting better. And like I said, 10x Genomics just released the platform. You can analyze 1 million cells at a time, which is a pretty big step forward, because currently we were capped about 80,000 cells at a time. So it's a pretty big leap. Another drawback is that the single cell data sets are a lot noisier than bulk RNA-seq data. There's some computational challenges to this. And one of the big things is that you lose spatial information. So I'll just briefly mention what many consider to be the next frontier of transcriptomic profiling, which is spatial transcriptomics. So this type of analysis allows you to capture gene expression information in the context of a tissue section. And you can superimpose your gene expression signatures onto a tissue histology section. And you can reveal spatial organization of cell types and biomarkers. So it's kind of like a digital histology, if you will. This is how this platform works. You have a slide here with a few different capture areas. And within this capture area, you have barcoded spots that look a lot like the gel beads we talked about for single cell RNA-seq and the multiome assay. So within each one of these spots, you have these oligos, and these oligos have the poly T that we talked about, the UMI, but then a spatial barcode, which is pretty much like a GPS coordinate that will allow you to map your RNA back to a specific area on the slide. So you can overlay a tissue section here, stain your tissue, lyse your tissue, and construct some RNA-seq libraries. And then you can map your RNA expression back onto the tissue and look at the RNA expression profiles within a tissue context. So this is kind of the results that you'll get from this experiment. Again, you have clusters of spots that represent different transcription profiles, and you can overlay these spots onto your histology section and look at the gene expression signatures there. So one of the things I didn't mention here is that it's not exactly single cell. The resolution here, you have some space in between these individual cells. So you're capturing multiple cells in an individual spot. So to kind of get closer to single cell, there have been some people in the single cell community that developed some methods to deconvolute these spots so you can try to figure out what types of cells you have captured by each spot. So this will allow you to look at like pie charts. The pie chart will tell you what proportion of the spot captured which type of cells, which can be important to getting closer to single cell resolution. But I think there's still some development that needs to happen here. So just to summarize what we talked about, the benefits of single cell RNA-seq over bulk analysis and how spatial transcriptomics can retain the spatial information, and using these both together, we can gain a lot more insight into the biology and answer some really important biological questions. And with that, I'll take some questions from you guys.
Awesome. Thank you, Mr. McSwiggen. If anyone who has questions would like to raise their hand, and I'll try my best to call on you in the order in which you raised your hands.
So maybe I'll ask, is there anybody in the class that is interested in bioinformatics or has programming experience? Devin, a little bit. A couple people. Yeah, so I think it's kind of an important skill, especially when you're a little bit younger, maybe able to pick it up a little bit quicker. But just there's a lot of people in the biology field that know a lot about biology, but it'd be a good idea to have more programming backgrounds so we can get more insight from large data sets, especially in the genomics field, which has extremely large data sets. And it's kind of hard to really answer questions that you want to answer without knowing some programming behind it.
I saw, was it Anoshka? Did you raise your hand? Did you have a question?
Oh, no, I was just saying that I was interested in bioinformatics because I did a small project on it last summer where I was trying to classify pathogens. And so I took pathogens and I took their DNA sequences and I basically converted it. I found a discrete Fourier transform of them and divided by the entropy to kind of create a metric for classification. And so I'm still working on the project, but it's basically about trying to find if there's a new virus, we're trying to classify it and we're trying to find the best way to classify it and trying to make a faster classification method.
Awesome. Thank you for that. Anyone else? Any more questions, comments? Yes, Naveen.
So I know you mentioned that it's good to have some programming experience in the fields of computational biology and whatnot. So my question is, if we don't have programming experience already, do you think it's possible to obtain that background in college, or how do you think the best way to go about it would be?
Yeah, so I think there are some college classes that will teach you a little bit about bioinformatics, but there's a lot of good boot camps and tutorials out there online that can really get you the information that you need to get you started there. I mean, if you're interested, you can contact, I don't know who your contact is, maybe Dr. Christina Wong or somebody, and ask them to send me an email and I can send you some online tutorials that may get you started. So in my experience, so I went to the University of Nevada, Reno, and they weren't, there's not that many bioinformatics classes. I think it's starting to pick up more now. There's a lot more need for it now. But I don't know. I think you can probably get some better education just from the internet. But I think it's a skill that can be picked up relatively easy. I mean, it's something that you need to practice and continue to do all the time to really get good at it. But yeah, if you want to send somebody an email, I can get you some links and some tutorials that may help you out.
Awesome. Thank you, Naveen. Okay, so we have a question in the chat box from Samuya. She was wondering how necessary the ability to code or program is in general when it comes to sciences in college. She heard that no matter where you go or what you get into, with some obvious exceptions, that you should know how to code, and I want to know if that is true.
Yeah, so I don't think it's imperative that you know how to code. I mean, I think one of the goals in genomics is to build more user-friendly desktop applications like I showed for Loop Browser. But I think it's a pretty important skill to have. And I think you can, I mean, for one, you can get some pretty good jobs. Everybody is looking for bioinformaticians. It is pretty decently, and it'll help you out. So, I mean, if you're interested, I think it's a good skill to try to pick up, but not necessary. Yeah, Devin.
Thank you for your presentation. It was really interesting. My question is kind of a bit of a clarifying question, I'd say. How does adding the barcode really work? How does that actually happen?
Yeah, so so you're attaching the poly A tail of the mRNA to that oligo DT, and then you use some enzymes that will pretty much attach to those areas and transcribe, so continue on along the mRNA, and just due to the basic complementary, it'll add those barcodes onto the end of your mRNA. Does that make sense?
Yeah, that makes so much sense. Thank you so much.
So yeah, we got another question in the chat box. The question reads, what is the difference between computational biology, systems and biology, and bioinformatics?
Yeah, that's a good question. So systems biology would be to put multiple different systems in the same context and try to figure out how a lot of these systems work interchangeably, like together, how these systems work together. Bioinformatics, I would say, is similar to computational biology, where you're just trying to gain insight from biological information. It's a good question. So I know you guys are all working on presentations. When is the ceremony? Is it tomorrow, Friday?
Friday.
On Friday? Okay. Cool. So if there's no other questions, maybe give you You have the rehearsal tomorrow. Yeah. If there's no other questions, maybe we can give you guys 20 minutes or so to 20 minutes extra to work on that. Yeah, of course.
Yeah, go for it.
Maybe typing your question out.
Yeah, I believe people are typing in the chat. And while people are doing so, I just want to give a healthy reminder for all of the students that education really needs your headshots for culmination ceremony. So if you could email education, I can resend the email down below. But if you haven't sent that headshot, please do so by the end of today, just so your photos can be put in the culmination ceremony and you can be recognized. Really appreciate it. Okay, question. So how did you get into the major you're in right now, and what made you realize that's where you wanted to be? Great question.
Yeah, that's a really good question. Um, so I think a lot of you are a lot more ahead of where I was in high school. So in high school, I wasn't really informed about PhD in medical school. So I kind of just bounced around. I started college as an engineer, moved over to neuroscience, got a little bit interested in medical school. So I went into molecular microbiology. And then after finished, and when I was in undergrad, I got into Dr. Yan's lab. And I just found the lab work to be really rewarding, really interesting. So I wanted to continue doing that. So then I got into a PhD program there. And it's pretty much where I am today. So I don't think it's really important to, I mean, for some people, it's important to identify what career path they want really early in their career or really early in college, but I don't think it's necessary. Dip your toes into a lot of fields, figure out what interests you the most. And that'll probably make a more interesting career for yourself.
Okay, was it hard to get into the lab? What was the process like, or was it just like you asked to work with them through email?
Yeah, so I was lucky enough to have a friend that worked at the university that knew Dr. Yan and kind of told them what my interests were. He introduced me to Dr. Yan. And so that's where I kind of got into his lab. But yeah, I mean, if you're looking to get into a lab, professors are always looking for undergraduates or other technicians or whatever you want to work in their lab. So yeah, if you're interested in the lab, just reach out to them, send them an email, tell them you want to work in the lab. Most of the time, it'll probably be okay. Sometimes it might be a little packed, but yeah, I mean, for me at the University of Nevada, PIs are always looking for graduate students or undergrads to work in their labs, so it's not that hard of a process. I'm not sure about UCLA or the colleges that you want to go to, but like I said, if you're interested in the lab, just send them an email.
Okay, how should college students get to know people that have contacts, or is it just through classes and clubs? Because I assume getting to know people at big colleges is hard.
Yeah, I mean, yeah, just try to involve yourself in a lot of activities that go on. Like, you can meet a lot of people through clubs, a lot of people through classes. There's a lot of social settings that go on in college that you can meet a lot of people through. There's also a lot of seminar series that colleges put on where there's a lot of college professors that come and talk about their research. Maybe go up to them after the class or send them an email afterward and say you thought their research was interesting, you want to work in their lab. But yeah, I mean, I wouldn't worry too much about finding contacts. I think it just immersing yourself in the experience. Along with that will come contacts that you can. Yeah. So, I mean, I'm kind of interested. Like, what are some of the colleges that you guys are looking at going into? Or are you at that process yet? UC schools? Yeah, that's true. There are a lot of colleges. I mean, for that, I would suggest just be looking at colleges, looking at some majors at the colleges that might interest you, and picking that way. Oxford would be awesome. Do you out of state schools? How is that? Yeah. Yeah, I mean, if you guys are interested, you can look at University of Nevada, Reno. It's pretty cheap. It's a decent education. Definitely for sure. Good for Oxford.
Awesome. So if no one has any more questions, I think we can call it wraps for today. But yeah, amazing lecture, Mr. McSwiggen. And it's great to, have conversation about, even things that are just unrelated to the topic and just about like everyone's college endeavors. As you're much more experienced than I am. I'm still in my undergraduate years. So it's good that you can give and apply your experiences to these students' questions. And then obviously, if anyone would like to reach out to Mr. McSwiggen, please feel free to do so. As you know, he has a plethora of resources and connections and would love to get in touch with any of you and all of you. And that goes for all of our previous lecturers as well. So yeah, thank you everyone for your participation and your
I
questions.
had one last question.
Oh yeah, no,
Um,
go for it. Go for it. We still got 10 minutes. Go for it.
well, yeah, I was wondering how can I get into contact with you? What's your contact info?
Yeah, so I'll put it in the
So
chat
you can
here.
put it in the chat box. That might be best. Yeah.
So just my first name dot last name at lumquist.org.
Thank you.
Yeah, of course.
Awesome. Okay, everyone. Thank you so much for attending. Thank you so much, Mr. McSwiggen, for your time and your energy. So yeah, everyone, take care. See you tomorrow at rehearsal at 1 p.m. All right,
Good luck
bye.
with all your
Thank you.
presentations.
Thank you.
so much.
Thank you.
Thank you.
Thank you so
Thank you
much.
so much.